Okay, thanks, Judy. I appreciate that a lot. The invitation, the introduction as well, and here on a Saturday morning. So I'm going to talk to you about what I do. And what I do is I'm a medicinal chemist, and medicinal chemists make drugs. So all the drugs that are dispensed in the pharmacy came from a medicinal chemist somewhere down the road. It's a long process, and we'll go through that as we go along. And then at the end, I'm just going to highlight a certain disease. And again, it's to just give you an idea of how hard it is to discover a drug. And especially in certain diseases where we really don't know why it happens, what happens. We are just trying to figure it out from animal models, translation to humans. So drug discovery. So historically, how did the FDA, how did approval of drugs come along? It's 120 years ago now, the first legislation, the Federal Food and Drug Act, that allowed basically just registration of anything. Someone said, okay, we think this works for this, and you could get it registered. But then they had an issue, and they kind of realized, well, not everything's safe. And there was a big controversy, 1938, diethylene glycol, which is antifreeze, which is solvent for sulfanilamide. They called it the elixir sulfanilamide, and sort of these carnival barkers, they go around, the proverbial snake oil salesman. And they were giving this out as a cure for things, and there was mass poisoning. Obviously, antifreeze is not something good. So in 1938, an amendment was passed to focus on at least safety. Can we at least know something's safe? Whether it works or not, that's a different question, but at least safety. And then that was about 30 years or so, and then they realized, well, maybe we need to know if something works too. So this Harris-Kefauver amendment required now to show efficacy. Does it work in the disease you say it does, and safety? And it was spurred by the thalidomide tragedy. I don't know if anyone is aware of the thalidomide tragedy. Thalidomide is a drug. It's actually still used right now. But what happened was it was a drug used for morning sickness. And the way chemicals are, compounds are, they are 3D molecules. And there are two separate versions of thalidomide. One of them was efficacious and worked. The other one actually caused horrific adverse events, and they produced what was known as thalidomide babies. So these babies were being born without limbs, without arms, without legs, and it was all due to that one pose of the chemical. And I can show you real quick. I'm an organic chemist, so I love organic chemistry. I'm going to spare you most of organic chemistry. But if you look at your hands are mirror images, but they cannot be superimposed. Right? You can't put them on top of each other, but they're mirror images. Those are called enantiomers, and that was all it was. It was one part of the molecule was just the opposite, but caused that massive difference. So 1962, we got here. Now we got to show efficacy and safety. And that has basically kept up until now. A drug has to go through significant safety trials, and then it has to go to show that it works. So why does it take so long, though, to discover a drug? Well, about 1 out of every 1,000 compounds, and a compound is I call it a pre-drug. That's something that my group makes. My lab is actually just right above here. I have 5 graduate students working in the lab right now. So out of every 1,000 compounds that gets made and gets tested, 1 really gets into what we call preclinical testing. Once it gets into humans, only 1 in 10 that starts a phase, human clinical trials, will actually get approval. So if you now calculate that back, about 1 in 10,000 compounds actually makes it to a drug. So this is a 10, 12-year process just to get it sometimes into clinical trials. And historical timelines is more than a 10-year range. And that 10-year is now about 15 or so, just again with a few more regulatory issues. And this is just, again, another pictorial. You have, 10,000 compounds. Stage 2, you have 250 compounds. Stage 3, 5 compounds in regulatory approval. And it just funneled down, and everything kind of circles back. So if something fails in this, it doesn't go forward. But if it goes forward and fails here, well, it kind of goes back. We learn what we did wrong, and we make changes to it. So it's an iterative process, always looking at, is it going to work, and is it safe? And the safety is really a real problem, because in our work, we're doing safety in, animals, a rat, a mouse, a dog, but none of them are humans. And humans have such variation in many things, enzymes in your body, that it's very challenging. So once we get it into a human, that's a whole new way that we have to look at it. So again, this is just kind of how we look at it. So, medicinal chemistry, this is me. Drug candidate, that would be sort of the pharmaceutical, and then preclinical drug, that's sort of when we start looking at the FDA. So does it work? How can it be delivered? So can it be a pill? Is it an IV solution? Is it inhaled drug? Those all have to do with sometimes the property of the drug, but also the disease, right? If it's an asthma indication, you want it in the lungs. So an inhaled might be the best way to do it. IV formulations, you see a lot of IV formulation. Those are really best in a hospital setting, right? You don't really have that capability at your home. Those are all things that we have to think about, and that changes how we me and others as a chemist think about those compounds. Toxicology safety, is it safe? And how do we know it's safe? And sometimes you don't know it's safe until it's approved and you start giving it to thousands and thousands of people. And then pharmaceuticals, can we make it? Can we get it in a pill form? Can we get it into a inhaled form? Those are all very challenging, almost engineering type of issues. So this is just, again, this is what I do. I identify something, we synthesize it, we make it in a lab, make sure it's pure. And then we test it, sort of this in vitro. You think about like a test tube testing, and then an in vivo, that'd be an animal. A lot of our work is in rats and mice. And sometimes we can engineer those animals to be more helpful. We can make, especially for mice, we can make humanized mice. So we actually take human DNA, put it in mice so that they kind of express a little bit more closer to humans. And then what does your body do to it? Does it absorb it? How does it distribute? What happens to it? And how does extraction and excretion? How does your body get rid of that drug? It's very important for us to know as well. And then, we have to be able to manufacture it at a cost that's, is reasonable. I won't even go into pricing of drugs. That's a whole different thing that I stay out of. Then you have to go into sometimes larger animal models, non-human primate. So my experience too is, before I was here at UNMC, I was at Vanderbilt in their department, but even before that I was in industry. So I spent 9 years in the pharmaceutical industry. And when you think about these animal models, one thing we were working on an asthma drug, and we actually had to go to a sheep model because their lungs are a lot larger. We couldn't really model the human in a mouse or a rat, just didn't give us the size. And then again, oral bioavailability, that's what we really want. We usually think about a drug as a pill. One day, you wake up in the morning, you take a pill, you wake up the next day. And that's good for patient compliance. If you start having every other day dosing, that's challenging. Twice a day dosing is a little bit better, but one day, one pill one day. And then you get here into regulatory submission. There's a lot of genetic toxicity assessments we do, acute and stuff. So if it's say an antibiotic where you're going to take something for 10 days, that's a whole different regulatory approval than if you're going to take something chronically every day for potentially the rest of your life, right? We have to really look at that. And that also goes to side effects. What can we manage? You know, you think of a cancer drug where you might lose your hair. You're probably not going to take that if it's an asthma drug, right? That's probably not going to be a side effect that we can tolerate. But for a cancer drug, maybe it is. And then there's a couple ways we look at this. So we can look at patient symptoms. There's a disease that has been diagnosed. Is there an existing drug already out there, either on the market or maybe one that failed in clinical trials in a different therapeutic disease? Can we use that for a new drug? A lot of doctors are allowed to prescribe off-label, so that's the way a lot of drugs are found to work in different things. If they see a paper that someone thought that this drug might work, so you can do it off-label and get to an improved drug. And this is how the COVID drugs were discovered. COVID drug Paxlovid especially was a drug that was approved in pretty rapid time frame, that whole 10 years I was talking about. But that was discovered actually many years ago when there was another SARS outbreak that just kind of didn't have the quote unquote the legs that this one did. They did a lot of research, and then that one wasn't nearly as efficacious. So then that research just kind of stayed on a shelf, and they were able to revive that real quick. And the other is a disease. We look at a biological system that affects that disease, and that's how we get drugs. And that's how today most drugs are made. Is some biologists, we have a lot over in the across campus. They look at diseases, they look at these biological pathways, and then we make new chemicals and we get a drug. Again, this circle here sometimes can be years and years. I tell my students you're going to fail about 95% of the time, which is sometimes hard to get students to join my lab when I tell them that, but they still do. So what is the clinical trial process? I think we all think of 3 stages. We have phase 1, phase 2, phase 3 clinical trials. There are actually 2 other ones. One is phase 0. So phase 0 is a first in human trials, but it's given at a very micro dose. So it's a subtherapeutic dose. So when you think of like a 10 when you're dispensing a pill of 10 milligrams, we would probably give that to a human in a 0.5 milligram. We just want to see what the body does to that drug. And that's very important. We want to know what the drug does to the body, but what your body does to the drug as well. So you give it to a small number of subjects, maybe 10 to 15, and you just want to know the agent's pharmacodynamics, that's the drug does to the body, and the pharmacokinetics, what the body does to the drug. Is the half-life, 3 days? Does it stay in the body for longer than maybe what we anticipated from the animal studies? Or does it have a half-life of 10 minutes? You think of like an Advil that you take maybe 4 times a day. That's because your body is metabolizing and excreting that drug. It just doesn't have a very long half-life, so you have to take it more times. So that's phase 0. Small amount of drug, healthy individuals. They're usually young, college-age kids that need money, and they come in and they volunteer. And then the second is again with healthy volunteers, but it's a little bit larger group, maybe under 100 people. And now we really want to determine safe doses. So again, you're going to take healthy volunteers. You don't want to take, people with the disease, because if you have a unknown event, you want a young, healthy individual that can sort of overcome any issue. And you look at safety, determine safe dose ranges, and begin to identify side effects. So what they'll do is they'll dose much higher than what they think. So again, going back to that 10 milligram tablet that you're prescribing or you're dosing out, you'll probably go to 500 milligrams just to see what happens if someone took more than what they were supposed to take. And you want to know where that top dose is. So what's the side effect when you go to a larger dose? Is it just nausea? Is it a rash? Or is it something even more serious? So that's phase 1. Again, small group. These are usually short-term studies, just to see. The phase 2 and the phase 3 are really the heart of the clinical trials. This is when you're testing a larger group of people. And phase 2, you're going for now, does it work? So these are now individuals with the disease that you're going to study. So we'll go back to asthma. Asthmatics that maybe meet a certain requirement. And that requirement sometimes is they're not on other drugs to alleviate that. They want to just say, does this drug work by itself? And then the gradual increase in this test group allows for more, less common side effects. So in that first 10 to 100 people that are healthy, you may not see a side effect, but you may see that side effect now when you start to get to people who have the disease. And once it passes that, it has to pass some sort of endpoint. When you make the clinical trials with the FDA, you have to tell them what your endpoint is. Is it, less acute flare-ups of asthma, and you'll have some sort of mark that you have to meet. Cancer, if you're thinking about a tumor, it would be shrinking of a tumor a certain size. And then you go to this phase 3. This is the largest and the longest of the phases. Now 1,000 to 3,000. These are a wider range of people. You want to not necessarily restrict those people because you're going to ultimately treat everyone with that disease. And now does it still work? A lot of times it'll work in a smaller population. You get to a larger population and that just kind of fizzles out. Some of that's statistics. They just, in a smaller versus a larger. And again, now you want to find all the common side effects and even more rare side effects. And this testing group, 1 to 3,000, can be up to 10,000 or more, depending on the disease that you have. And I'll talk about brain diseases, and that's predominantly where I work, because we have a placebo effect. So you have to have a large enough number of people to have statistics above that. But for the most part, it's in this low thousands. And this can be challenging in and of itself, is to find 3,000 people with that disease that's willing to be in your clinical trial. So it sometimes can take a year or more just to find that many people. And that's why clinical trial is not at a single hospital. Think about Nebraska, we have a very small population. There's no way we would be able to find that many people. So we might have 30 of those people. A hospital in Dallas might have 400 or 500. So it's throughout the country. You also want to do that too, just to make sure there's no genetic issue. Sometimes in certain areas of the country, you have a lineage of maybe a Eastern European or something. You want to make sure you have a wide range of individuals. And that's the other reason we don't do too many clinical trials overseas either, because diet and everything else would be very different over there than what they would be here. And then the last is phase 4, and this is just safety during sales, post-marketing evaluation. A lot of times when drugs get a black box warning for a particular off-target, that's noticed in phase 4, because now you are just opening it up. A lot of people are taking it. I don't know if you're familiar with the black box warning, but when you see it on a pill box, it literally is a black box, and it has a warning on it, of whatever that might be, heart, say liver damage, and that's done here. And when compounds or drugs are pulled off the market, that's usually realized here after they've been on the market for a little while. They start seeing these issues. So that's clinical trials. Phase 1, 2, and 3 are the major clinical trials, and they will take 6 years, maybe. Part of that is recruiting subjects. Part of that is once the clinical trial's over, trying to look at the results, talking with the FDA, convincing the FDA that you met your goal, going to the next round as well. And most of these are blinded. You know, you hear double-blinded clinical trials. That means the patient taking the drug doesn't know what they have, whether it's the drug, placebo, and also the administrator, the doctor, does not know who gets what. So both parties are blinded. You come back and then you do the analysis afterwards. All right, so we're going to get into sort of CNS is central nervous system, so brain disorders. And like I said, I've worked in this area for all 20 plus years of my career. Right now I work in Parkinson's disease. We have a program looking at drugs to alleviate Parkinson's. I work in non-opioid pain area, and I've also worked in Alzheimer's. Central nervous system is even worse than regular drug discovery, so it takes even longer. So the overall clinical approval success, about 13% for non-CNS. I said that 1 in 10, 10 go in, 1 comes out, so about 10%. We're at about 6%. So less success, and I'll get into why that is. In this 3-year period, the overall success was 5%. It takes longer. Just the approval phase, so once everything's done and you go to the FDA, it takes about 20 months for that because of the way clinical trials are versus non-brain disorders. And there are 2 main reasons. One is we really don't have an idea of what's going on in most diseases of the brain. So a lack of understanding. Alzheimer's, we really have no idea what causes Alzheimer's. We have theories of Alzheimer's, but that's sort of after the fact. There are a few diseases, Parkinson's is the one I work in, that we do have a really good idea of what causes, or at least in the brain, what Parkinson's is. We don't really know the causes. There are some thoughts on that. And here in Nebraska, we have, I think, one of the highest, if not the highest percent of per capita of Parkinson's disease in the country. And more rural areas have a higher percent of Parkinson's than urban. So there is a thought of a pesticide, herbicide, groundwater from farming, but that's still not been fully proven. But the lack of understanding, but when we do understand it, your brain is so good at compensating. So if something happens in your brain, some other part takes over. So by the time you present, say with Parkinson's disease, you've lost 80% of that part of your brain. So trying to treat something where you've lost that much is very challenging. And then the next is hard to establish these relevant animal models. And I'll talk about that. Animals are not humans. They just don't have the same diseases that we do. And why is it so difficult? Well, the brain is just so complex. We have an idea of where things in the brain work, like what part controls vision, what part controls speech. But everything is so interconnected that, like I said, one part of the brain can be failing and other parts will come and take over. So I teach a couple of different classes, and it's just, I found this, I thought it was good. That it's just, there's so many things going on that we have a hard time understanding, and all of the things that we researched before, rats, mice, don't have anywhere near this complexity. So real quick, what are drugs? I'm sure everyone here, I don't have to tell you what a drug is. A drug is a substance to prevent or treat a disease. You can also use it to make diagnosis. They're designed to act in a certain site of action for a certain pharmacological target, and that's for the most part. We know what drugs, what part of the body it's affecting. But they can act as poisons if taken in excess, right? You take too much, you have a lot of overdose of things. So we balance this specific beneficial action with these side effects. So where do drugs come from? We get drugs from a lot of different sources. One is we get drugs from plants. So Taxol, one of the most used cancer drugs over the last 25, 30 years, comes from the bark of the Pacific yew tree. And here's the yew tree, someone peeling that off. However, when they were doing the first clinical trials, they realized that to get enough Taxol from this, you were going to have to wipe out the entire Pacific yew tree population just to run the clinical trial. So chemists came to the rescue and said, we need to know how to make this compound. It's pretty complex. You need to be a chemist to know that. And what they did was they found this compound. It's called diacetylbaccatin. And if you look, everything is there except for this part. And the nice thing about this is found in the yew needles. So when the tree drops its needles, we can harvest that. We can get as much of this and a lot more than we can of this. And it's renewable, right? Every year, new needles drop. So then a chemist down at Florida State found this, was able to make Taxol. Now we have enough supply for everyone. It's also produced by microbial fermentation, but that's, again, a much longer process. So Taxol is used in treatment of ovarian, breast, lung cancer, Kaposi sarcoma as well. It is a really, probably one of the most prescribed cancer drugs. We also get them from animals. So this is a story that I think a lot of people know now, but didn't even 6 years ago. So this is the Gila monster. And someone was looking at the Gila monster, had a grant from the NIH, and wanted to know, why did it only eat once every 6 months? And they isolated this, took the saliva, isolated this. It's a naturally occurring 39 amino acid. It's called a GLP-1. It's an agonist, and that is now exenatide. It's also basically Wegovy, Ozempic, Zepbound. All of those are variations of this. But this was just because someone, scientists, wanted to know why. Why does this animal only eat? And what GLP-1 does is in the brain, it promotes satiety, so you feel full, reduces appetite. In the stomach, it lowers gastric motility and gastric emptying, so you stay full longer. And in the pancreas, it increases insulin and decreases glucagon. So it's also not only for potential weight loss, but also for diabetes. And what this looks like, so this is GLP-1, and these single letters are amino acids. They are the single letter code for every amino acid. I do not know them all, so I'm not ever expect anyone. I know H, that's histidine. This is GLP-1. And then this is exenatide, or Bietta. That was the first drug that was in this class. And you can see basically where the green is the changes they made. And the reason they made these changes, just so your body, it'll circulate longer in the body. And then Ozempic, semaglutide, is basically this same GLP-1. Two changes here, but then they added this chemical here at the end. And again, just to allow for easier dosing, your body doesn't break it apart as fast as GLP-1. So all of these amino acids, your body is very good at breaking down these called peptides. So that's how another drug was found. And then here are some other examples of what drugs look like. So acetaminophen has been in the news recently. So that's here. I'll say without comment. The next is Paxlovid. So Paxlovid is the COVID-19. And you can just look at the structure. Again, just appreciating, even without being a chemist, look, this is really more complex than this. And it's a completely different part of the body, different receptor, everything. And then sequinavir is an HIV reverse transcriptase inhibitor. Again, simple, complex, very complex. And sequinavir, you talk about drugs and how there's a lot of talk about, pharmaceutical industry doesn't want to cure disease. They just want to, keep people on drugs. Sequinavir is one of the most successful drugs in the history of the world. And does it cure HIV? No. But it makes a death sentence a manageable chronic disease that people can live a full and normal life because of the discovery of this drug. And it's used with multiple, HIV is a cocktail, but this is the first one that really made that. And I teach antivirals in the PharmD and the P2, and I show a graph, and it was something like the rate of HIV deaths was going straight up, and once this drug came on, it almost straight down. So drugs from synthesis, when I'm up in my lab, and I will admit I don't do any chemistry anymore, my students do all the work, I just kind of think and help them along the way. So before a drug, this is sort of how your body works. You have this biological system, and you have all these different natural substances your body has. It comes in, it nestles in, it either turns it on, turns it off, everything's working just fine. Then sometimes your body starts working too much. Something's overactive, and we need to turn it off. And that's where we go here. We make a drug that fits into this receptor site and blocks the natural substance. So now we can turn that off. And that's a lot in cancers, right? A lot of overactive, cell proliferation. We want to inhibit that. So we make these kind of drugs. And you block that, and then you block that tumor from growing. Or it's not working enough. So this arrow's a lot smaller here, and we need to turn that on, and that's called an agonist. So we designed something else to fit in here, better than what the natural substance does, and you turn it on, and now it's working the way it should. So that's sort of how we think about it. The other analogy I like to do is think of a lock and a key. You have these keys here are the drugs. The lock is the biological system that we want it to either unlock or lock. And we have to make these to fit this perfectly. And then we start here, the initial building blocks. These are all the chemicals we use to synthesize these, and you can just see the colors. You can put them together in different variations. And we see, does it work? And I like to, if you have a lock and your key doesn't fit really well and you kind of have to jiggle it around, but it will work ultimately. We want that perfect lock where you put the key in, it turns it, and it has no issue. And that's why it takes really a long time to get there. So the last few minutes I have, I'm going to go into a specific disease and just kind of go over how we think about it. So the major disorders of the central nervous system broke them down into kind of a couple different categories. Mental health, so we have depression, psychosis, anxiety. And then neurodegeneration. These are really the big Alzheimer's, Parkinson's. These are the two that are really going to expand as the population gets older. Huntington's disease. This is one that's been in the news recently as well. They had the first dose of a genetic manipulation, I think it was in London, for Huntington's disease. Huntington's disease is a death sentence. If you have the Huntington gene, you are going to get it. It's 100%. So this is really exciting. It's not curative, at least not yet, but at least as a treatment. And then ALS, multiple sclerosis. And then we do have a couple others. Addiction is a disease, but it doesn't fit into either of those. You have infections and cancers, right? Glioblastomas. I want to talk about schizophrenia. Schizophrenia, it's from the Greek, basically means split mind, defined by a German psychiatrist more than 100 years ago, and then a Swiss psychiatrist, Bleuler, coined the term. It's a thought disorder. It's illogical thinking. It's lack of reasoning. It's an inability to recognize reality. It's one of the reasons why it takes so challenging to treat is no other animal has that, right? So we have to really think of how we can translate. The symptoms are very variable for each individual. But the disturbance of perceptions, these hallucinations, are the most common. And the most common of those are the auditory, so hearing voices. It's a chronic condition. There's drugs, environmental toxins can cause acute psychosis. About 30% of the people with schizophrenia will spend a significant portion of their lives in mental hospitals, and that's even with therapy. About 1 to 2% of the population will suffer. So it's a small percentage, but that's still a large number. And then 2 to 3% will suffer from less severe symptoms that don't meet that diagnostic criteria. So there's no definitive diagnosis. You can't come in and do a blood test and say that you have schizophrenia. And they're broken into 2 different classifications. There's positive and negative symptoms. So positive is you have too much, and that's the delusions, hallucinations, the bizarre behavior. That's where most of the drugs, antipsychotics, treat the positive symptoms. Treat, and I'm going to put those in quotations because I don't think they treat very well, but they do, they are marketed drugs. And then the negative, the too little, social withdrawal, intellectual impairment, this avolition, this loss of initiative and motivation, this anhedonia, this just inability to derive pleasure in life. Those are the 2, and the negative symptoms are something that really no drug treats. So we can usually, if you take your antipsychotic, those hallucinations will go away, but you don't have a way to bring those individuals kind of back to society. And I like showing this, and I teach this class from our high school classes up to college. This is an artist, Brian Charnley in the US, or in Europe, who had schizophrenia, and he decided to take himself off his medication and chart his journey with self-portraits. So this is an initial self-portrait. This is what he looks like. So after taking it off, so April 20th, he started to cut back. And then you can see these are all, in his mind as an artist, what self-portrait. And then you can see as it goes along, now he's not on the drugs at all. July 19th was his last self-portrait. Unfortunately, 2 or 3 days later, he committed suicide. So this is just, again, just a slice of one individual's with schizophrenia's mind and how they're thinking. And it's interesting that someone as creative as him could track that. We don't have that in many others. So how do we treat and how do we research this? Well, prior to drug treatment, there are a few options. You're restrained in hospitals, you have shock treatment or insulin-induced seizures, prefrontal lobotomy. Some of you may know, but that's a pretty gruesome, right? It's a needle through the eye into the lobotomy. Since the 1950s, hospitalizations began to decline, and that all coincided with this drug, chlorpromazine or Thorazine. That was the first antipsychotic. And unfortunately, because of societal changes, once this drug came out, then sort of, states and national support for hospitalizations really declined. Like, well, we have this drug, we don't need to pay for these hospitals anymore. So we have 2 types. We have a typical and an atypical. Typical, we're the first generation, again, mainly treating these positive symptoms. But one of the real issues in the side effects in these, they have these movement disorders due to excessive D2 is a dopamine receptor. So this was a major issue with this. People would take these drugs, they would start feeling better, and then they would have these side effects. So then researchers developed these, a second generation, clozapine and risperidone are 2 of the major ones. They treat, again, the positive. They say they treat some negatives. I'm not a clinician. I'm simply a researcher, but I don't think they treat many of those negative symptoms, at least from the results I've seen. However, the major side effect of these are hyperglycemia, diabetes, and that leads to excessive weight gain. Some of these individuals will gain 100 pounds in 6 months. So they take these drugs, they start feeling better because they don't have the hallucinations, but this weight gain, so they stop taking it. And it's this real vicious cycle. So schizophrenia is a thought disorder, unique to humans, extremely difficult to develop a model for us to do preclinically. You know, the question, can we give an animal schizophrenia? The answer is no, but we have learned some things. So amphetamine and cocaine use cause psychosis in humans. You get these paranoid delusions, you get these stereotyped compulsive behaviors, just, you know, as you think of an OCD on you know, sort of steroids. And you also have hallucinations. Patients with schizophrenia take amphetamine, see their symptoms worsen. So, you know, the thought was, well, what happens if we use amphetamine as a model for schizophrenia in an animal? So when we give animals amphetamines, you have these same stereotyped behaviors. You have sniffing, licking, gnawing. That's similar to this repetitive. Significant hyperlocomotion, they really get ramped up. So these are, again, we use this as a screening device for antipsychotics, thinking that some of these behaviors will translate. But it's not schizophrenia. And translation's not 100%, but all those drugs I just showed you, the typical and atypical, work here, but it just works in this model. So how does this work? So we have this little box here, and in this you can't see, there's cross laser beams, both going this way and this way. And we put the animal in there, with and without amphetamine, see how much they move around, see what that hyperlocomotion is like. So this is called an open field chamber. We vehicle versus amphetamine, and we just measure how many laser beam breaks are there, just as an indication of how much they're moving. And you can see here, you put them in there. They have a lot of beam breaks, but they settle down. You know, animals want to sniff out. So once they get in this sort of area, we give them amphetamine. And you can see this red and orange is amphetamine. You can see how much they significantly start moving. And then we give a drug. And this is in a dose, so it's a 3, a 10, a 30 milligram. And you can see we can reduce that movement. I mean, we're reducing the amphetamine induced, but I don't know that's schizophrenia. And the black here is just if you just give it saline. So we're kind of down to normal. So that's one way we test things. The other thing we notice is schizophrenias have difficulty screening out irrelevant information. It's really a sensory overload. And one of the things is what's called pre-pulse inhibition. So they do not have this. So if I'm, you know, July 4th, let's say, right? And I'm sitting in my living room and I know my neighbor's gonna blast that firework and I don't know when, and it blasts and it scares the living you know what out of me. But if he put a little firecracker first and then did that, that's the pre-pulse. So I would be trained, okay, I heard that, now I'm not gonna be nearly as startled. Schizophrenics don't have that. They would not be able to do that. And rats on amphetamine also don't have that. So we do that. We try a pre-pulse. So we give a pulse, normal response. If you give a pre-pulse and then the pulse, this is an inhibited because they're trained to do that. And we put them in this little area, soundproof, and we test that. We pulse and a pre-pulse. And you can see sort of this bar, there's a reversal of that. Again, it's not schizophrenia, but drugs have worked, and this is why it is so challenging to do. And the other is when, at the beginning, I was talking about a placebo effect, especially for antidepressives and anti-schizophrenic drugs. When those individuals start taking a drug, they do start usually reporting better symptoms, even if it's placebo. So you have to have a really large number of individuals to filter out that placebo effect. So I still think drug discovery in schizophrenia remains a major unmet medical need. I think in where we are with payers and insurance, it's going to be very challenging to introduce a brand new schizophrenic drug because insurers are going to say, well, we have these that work. But I don't think they work. So we still are working at it. The existing drugs have significant side effects. That's even if we could get that down would be good. And then the major challenge is this lack of translation, animals to humans. So that's kind of what I wanted to talk to you about.